It's my great pleasure to introduce today's first plenary speaker. That's Maryann Martone. Maryann got her PhD at UCSD in the mid-1980s, and she stayed on there as a postdoc with Mark Ellisman and then joined the faculty and has stayed there firmly rooted ever since. She trained as a cellular neuroscientist and neuroanatomist, but in the 1990s, she began a foray that has been very significant, moving into the area of neuroinformatics. I got to know her then through a collaboration on a NPACI NSF-funded supercomputer project, and have followed her evolution as into a leading neuroanatomist and neuroinformatician over the ensuing years. Maryann's contributions have included the establishment of the now highly successful cell-centered database, and additionally she's had an extremely important role in a number of international efforts, particularly the Neuroscience Information Framework, which she is currently the PI for and has helped shape that and drive that to a very important enterprise that we'll be hearing about this afternoon. So without further delay, because we must stay on time and get everyone to the ship on time at 6 o'clock, it's my wonderful pleasure to introduce an individual I greatly respect, Maryann Martone.
Thank you very much, David, and thank you very much for the organizers for inviting me to speak at this wonderful conference. Kobe has been an absolutely spectacular venue. I'm going to talk today about the Neuroscience Information Framework project, but I've entitled my talk The Possibility and Probability of Establishing a Global Neuroscience Information Framework. I entitled it that because even though we can run projects like the NIF project, as I call it, this really is a community effort, and there's no single group or single strategy for being able to knit all of the wonderful diversity of neuroscience data and tools that we've seen presented at this conference and others into a framework without community cooperation. So I hope today to try to convince you that such a framework is possible, and then also work to how we might make it a little bit more probable that we would likely achieve success. So I started the slide with just sort of considering all the different ways we access data, and over the years that has been mainly in the form of books, indeed for centuries since the invention of books and then also the invention of the printing press. If we wanted data, we went and we looked it up and we tried to use it. Obviously, for the amount of data that we're producing now, all the gene sequence data, that's not a very efficient way of doing that. And sometime around the 1980s, when I started my academic career, we began to have personal computers, and we could have things like spreadsheets, and we could put that data in spreadsheets, and we could much more easily operate on top of it. We didn't have to look it up in books. We could actually use it and use some of the basic search mechanisms. In the early 90s, I remember very well using Mosaic and some of the very early internet browsers, because it now became possible to post this data on the web. And we had ways of exchanging documents on the web, we had informational pages on the web, and we had databases that were also available through the web that you could go and find out where that database was and then get into that information content. There's been a large push lately for transforming this idea of the web into what Tim Berners-Lee calls the web of data. The idea now that we're not just going to use the web for finding things that have data in them, but the data themselves will be on the web. There's various terms that are used: linked data, open data, semantic web, what have you, but the basic idea, again, is that all the data elements that we produce are going to be linked into one of these very, very large, gigantic graphs that we're somehow going to use. Well, this leads to what I call the mountain of data problem, right? So what would you like to be able to do with this large amount of data? If you can access the world, how do you find what you're looking for? And what would be the types of things that you would like to find? Well, you'd certainly like to be able to go in there and find what is known. And you'd like to be able to ask questions like, what's the average diameter of a Purkinje neuron? And is GRIM1 expressed in the cerebral cortex? Or what are the projections of the hippocampus? And I want to make it quite clear that when I want to ask these questions, what I'm really asking at is, where's all the data that is relevant to this? Okay? Because people have made various measurements, I'd like to be able to make sure that I could get all those measurements and actually look at them and make use of them. That's what I'm talking about, not getting a yes or no answer. But more importantly, of course, we'd like to know what is not known. We'd like to be able to discover new things by looking at the connections amongst data. That's always what we've been doing. We look and we see different ways. That's why we have conferences. That's why we have books. That's why we have papers. We'd also like to know where in that gigantic graph and mountain of data are the gaps in knowledge. What don't we know? Right now, it's very difficult to find this because when we ask a question, we can't even pull out what we do know, so how do we know if we don't know it, or how do we know that nobody knows it? And it turns out that even though you can make those nice graphs, and Tim Berners-Lee can get up there and wave his hands, in fact, trying to build a system that makes use of this mountains of data takes a whole lot of effort, that there's a whole lot of things that need to go into that. It's not just that you put your data on the web, but you have to think about how people are going to query it. What sort of strategies are they going to use to rank what they have? How are they going to understand the things that they get back? How do they know whether to trust it? How can they bring it back to the provenance? What sort of infrastructure do you need? You can query a gigantic graph of data, but nobody wants to wait 8 days for it to come back, so you're going to have to be able to index it. So you need some way of being able to gain this rapidly. You'd like to be able to hook analysis tools. You also have to recognize that there's been a whole investment. We haven't been sitting there twiddling our thumbs waiting for the semantic web to come into existence. So there's a whole lot of data there that has to be transformed and made available. How does that happen? We'd like to create tools to create that when we create new data, it's much more ready to be linked. So in fact, it's a gigantic problem, and that's why, one of the reasons that I'm here today. So we've all known, and we've heard this mentioned, Stephen showed this yesterday, that in fact when you acquire data within a framework, that is when there's some structure to it that we all understand, that it's very effective for actually organizing data. We've heard Google Earth, we've heard Wikipedia, and we've recognized that there's really two good ways of doing it. This is not the only way, but there's knowledge in space and spatial relationships. Where does the data come from? Generally, when you talk to a neuroscientist, for example, if they reference the brain, the first thing they say is, where in the brain did I get this data from? And the second is knowledge in words, terminologies, logical relationships. What are you talking about? And we communicate about this, and we communicate about this in terms of concepts and other sorts of things. And what I'm going to argue is that even though these are very powerful frameworks and that they're very difficult to implement for the brain, it's really what we have. We don't have a common data type. A lot of people would like to all standardize around data types, and whereas bioinformatics has done that effectively with the gene sequence, we generate a whole lot of different data types, and those different data types are in different formats. It's very difficult to sort of define something that's common across all of neuroscience. But again, the power of this is that there are algorithms, there's all kinds of clever things that are being done by computer scientists and others once you have the data in a framework. We've heard a lot about assembling data into coherent models. That's what we've been hearing about yesterday. We've been hearing about that today. And there's a very beautiful paper which shows you can take a whole bunch of messy data, photos on Flickr that are very messily annotated, and you can actually use all these different points of views to construct things that you know about. So we have the Roman Colosseum, we have the Matterhorn, we have the Statue of Liberty. So when you have data in some sort of framework, you can actually extract interesting data from it. And we've seen that there are efforts underway using a similar framework which says, listen, I know the where of it. You saw that yesterday in Stephen Larson's presentation, and he's giving a demonstration out there called the Whole Brain Catalog, which says, hey, I can find a way of spatially indexing all of the data there is about a mouse brain so that I can bring it together. But I'd like you to sort of consider a what if. Okay, so what if it turned out in our nice little photo experiment that the Matterhorn could be 15 different things? What if it turned out there were 6 billion Matterhorns, all of them somewhat similar, but all of them different from one another? What if the Roman Colosseum was called by 45 different names? What if the photos that you saw represented 1/1,000,000th of the whole with no context whatsoever? Where does this belong? Is that part of the Statue of Liberty, the Matterhorn, or the Roman Colosseum? I don't know. What if those photos weren't annotated at all, or what if they were tagged with something called 1 or MM45? Would you know what to do with it then? Does that have any meaning to you? And what if the Statue of Liberty was represented not as a picture but as a mathematical equation or a scatter plot? Would you be able to find it? So I'm sort of asking people to step back and think not about the own wonderful data that they produce and their own wonderful world of databases that they have, but if you were trying to query across all of this, and this were the situation, which is pretty much what is true for neuroscience, how would you go about finding this? What would you do? So the Neuroscience Information Framework Project, or NIF, that David mentioned, was actually supported by the NIH Blueprint. That's the consortium of the U.S. National Institutes of Health, the Neural Institutes. They sponsored this project because they said, hey, we've been investing in a whole lot of resources. We actually don't even know how many are out there. We don't even know what we funded. We'd like to know how many there are, what type are they, and more importantly, how can we use them? How can we make people aware of them? How can we find them? So the NIF project has had to tackle all of the issues that I mentioned before. We've set up a portal that can be used to find and use neuroscience resources. We've tried to create a consistent framework for describing resources, and by resources I mean data, tools, materials. As I'll show you, it's supported by an expansive ontology for neuroscience, and it's had to employ technologies to search those silos, those databases that are currently not well indexed by search engines like Google. So NIF essentially provides simultaneous access to multiple sources of information through what I'll show you as we call a concept-based interface. It provides very simplified and neuroscience-centered views of very complicated resources to make them understandable. But the point that I'm mostly going to talk about today is we've done this, and we've done it reasonably well, but we haven't done it perfectly. There's a lot to be done. What the NIF project, I think, has really begun to show is that when you actually try to integrate all these resources, when you try to figure out what's in them, we have gained a wealth of real information on practical issues of how you can search across and make use of all of this mountain of data. So if you want to implement this sort of web of data, what would be required? The first thing you need to do is know what you've got. So the first thing I want to point out is how many resources are there. Again, that was NIF's original mandate. So we went out and we've looked on the web, and we go to conferences, and we use search engines, and we look at the literature, and we look at all of these things, and we keep finding new sources every single day. We've been at this for several years. You'd think we would have maxed out by now, but we haven't. We currently, in what we call the NIF catalog or NIF registry, have about 3,000 different resources described. Of those, 2,000 of them are databases. We have a backlog of another 4,000 that are awaiting curation that haven't yet been done. The curation involves applying keywords, trying to figure out what these things are. And we find more every day. I find a lot out here when I come to these conferences. So the state of finding resources is very difficult, but the main thing is that there's too many databases to visit. You cannot, as a research scientist, go to all of those databases, and you don't necessarily know where all the things that you would like to find out are stored. They may be in a database that you know about, but they may be someplace else, and ideally you would like to know what that is. So NIF provides something called the NIF Data Federation, where we essentially say you can issue one query from the NIF portal, but we're going to try to search across as many of these as possible. And we use a variety of different technologies and techniques to allow that to happen, because we have to recognize that we need to work with resources as they exist, not the ones that are yet to come online. So currently NIF offers access to about 50 different databases that have combined over 26 million records. We organize this into a framework. In our case, the framework is a level of nervous system and also data type. We have to provide a way of searching it, so we have a keyword query interface, because if you're searching across 26 million things that you don't know about, the first thing you're going to do is do a probe like you would with Google. Our goal is not to usurp these resources, but actually to link people to them, so once you get there, you actually go to the resource. And as much as possible, we try to look and say, what's inside of these? How can we make this comprehensible? And is there some uniformity that we can impose? So this shows the current NIF query interface. And when you issue a query up here called hippocampus, the first thing you notice is that NIF expands that into things that we know actually mean the same thing. So NIF takes care of synonyms. It then provides information in different sources. You have the NIF Data Federation and the registry, which we talked about, but it also has its own web index, it has a literature index, and it searches through grants that were funded through NIH, which somehow rated its own tab because they're the ones who are actually funding us. And over here on the left, it sort of dynamically organizes things according to the data type. So is it cell data? Is it software? What is it that you're looking at? And NIF has several features to try to make this understandable to people. So again, we provide interesting queries, expansions to go and see what we're looking at. We categorize data. We provide links back to the original source. We simplify the view to try to take the essence of what a very complicated database is. And we provide tutorials on how to use the resource, because all of these things are different. So what has NIF learned when we've actually gone through and tried to use these things? Ideally, again, we've now got 26 million records. You'd like to be able to ask questions and get answers. So a reasonable question is the gene GRIM1 expressed in cerebral cortex? If you do that, NIF actually says, well, I've got three sources, the Allen Brain Atlas, GenSat, and the Mouse Genome Database, that provide me information on a string that says cerebral cortex. One of them says 23, one of them says weak signal, and the other said actually the number 1, which we found out when we went into the resource actually meant true. Okay? So this shows you several well-known issues in search. Even though we took care of the problem of searching across multiple different databases, we still have difficulty finding data. We know that there's inconsistent and sparse annotation of scientific data. We know there are many different names for the same thing. We know the same name means many different things. And we found a wealth of what we called hidden semantics. So you'd often find 23. Does that mean it's present or not present? You had to go into the source resource, try to figure out what they meant by 23. You had to go in and figure out what weak signal meant. You had to go figure out what the number 1 meant. In this case, it was true. There was a lot of translation that had to go on. Worse, if you actually went in and said, well, what do they mean by cerebral cortex? You found, well, some people would tell us, and what they told us was not always consistent. It wasn't always clear that that territory, cerebral cortex, pointed to the same substructures. Sometimes you simply couldn't find it at all, and sometimes you found it and you found a whole bunch of different structures. So even though we found the string cerebral cortex, we really weren't sure what we were actually looking at. We also found that when you went into these resources, you wanted to be able to ask a question, or what are the connections of the hippocampus? This shows you the interface from the COCOMAC database, and this is the Brain Architecture Management System. Both of these are extremely rich databases. They're also constructed completely differently. It's very difficult to know from just looking at this whether there are connections or whether there are not connections. When you went and you looked at the structure, you found that there were many different ways of saying connected to, and every single one of them that we could possibly think of was used at least once, and sometimes they were used, there were other ones, again, that you didn't even know about. And ideally, again, when you actually sort of thought about it and you looked across all of that, that they were saying some very simple things. There was a database, they were talking about connectivity from, connectivity to, there was projection strength, and there was evidence. So doesn't this look a little bit more coherent than all of the different data sources that we have to go after? Databases, of course, are not the only place where there are things. There's a lot of things that are reported in the literature, and one of NIF's goals was to be able to go to the literature and find out what resources are presented there. So again, we had a nice use case. I want to know what studies used my monoclonal antibody against GAD in humans. We went and we looked at papers. We used automated text finding algorithms and other sorts of things to find it, and then we went in and we validated. Much to our surprise, or perhaps not to our surprise, it was actually fairly difficult to track down reagents in a given paper. A lot of papers said, well, I used a reagent according to this study, and you went there and they said, and I used it according to another study, so you had to go track it back several different papers. Sometimes they told us about the reagent, but they didn't give us the vendor, so we didn't really know whether it was our antibody or not. Sometimes they gave us the vendor, but the most frequent piece of information they gave us with vendor was the physical location. So they would say this was Sigma St. Louis, Missouri, which is a fairly useless piece of information right now, but they wouldn't tell us the catalog number. And when you went and tried to track down the reagent from Sigma, it turns out they've got 5 of these. And so you really don't know which one they use. They're all different sequences and things. So it turns out to be very, very difficult to actually track this information down because it's not available. So, what is the result of this? The result of this is that searching and navigating across individual resources takes an inordinate amount of human effort. We have a full-time curator. We spend a lot of time trying to figure out what these resources are. And what we've really concluded is that we're not publishing data in a form that makes it particularly easy to integrate. We publish data for local resources that are meant to be consumed by a few people, and we don't make what we mean clear to a search engine or even to a human sometimes. We use many different data structures to say the same thing, and oftentimes we don't provide crucial information in the first place because, again, we know our data very well and we don't assume anything's going to care. So, NIF has basically said, listen, if you really want to be able to use shared data and put linked data out on the web, there's several things that have to happen. A, you have to be able to find it. So it has to be accessible through the web. You need enough annotations, and you should structure it or semi-structure it so you can find it. B, you have to be able to use it. You should specify the data type, so you should know what you're looking at, and you should be able to open it. It needs to be a format that's not proprietary. And C, you have to know what the data mean. And part of that are semantics. Part of that is context and experimental metadata. But again, we see a whole lot of symbolic notation, custom notation that makes it almost impossible. So it was absolutely necessary that NIF turn to something that we've heard dismissed quite a bit and discussed quite a bit, and that is ontology. And ontology, as we have heard, is essentially an explicit formal representation of concepts and the relationships amongst them that defines a domain. Even though it's originally a branch of philosophy, and it means literally a theory what is, its main point these days is that we can express human knowledge in a machine-readable form. So you may have something that looks like this, that brain has a part cerebellum and a Purkinje cell is a neuron. There are many ontologies in use, some of them that are very widely used, for example, the gene ontology. Now, when I talk about ontology to various different communities, I usually get one of two responses. When I talk to computer scientists, they consider it a completely dismissive sort of thing. They typically say, you know what, I'll say you need to use ontologies, and they're like, yeah, ontologies. They don't think about it in terms of data integration, they think about it of explaining what is in their own particular resource. But even worse, when I talk to neuroscientists or journal editors, I see this Gary Larson cartoon. No, well, Fluffy, you've clawed the furniture for the last time, I'll not tolerate this. And what they hear is usually completely A, I don't care, or B, active hostility towards what it is that you're saying. And we actually heard some of this the last couple of days. However, what I'm hoping to do here is to show that ontology can do a lot for us when we're trying to find data, when we're trying to deal with this mountain of data problem. So what can it do for us? It can express neuroscience concepts in a way that are machine readable, okay? It focuses on definitions, providing meaning that allows us to be able to use this not by what we call it, but by what it means. We can encode rules by which a class is defined, and those rules need not be particularly profound. For example, a GABAergic neuron is a neuron that releases GABA as a neurotransmitter. That is not highly controversial. It allows us to form properties. It allows us to put quantities. But the main thing that I want to emphasize is that it provides the universals. It's the universal knowledge, the language that we use to communicate across resources. That's what allows us to take a data structure that's very proprietary, normalized, and difficult to see, and says, out of all of this, these are the things I'm trying to communicate to you. So we find that, for example, it provides the basis for doing concept-based queries to probe and mine data. I'm not going to talk about this last point, but I'll be happy to do it on the social or any other time, and want to say that as a branch of philosophy, it actually also makes us consider very strongly what it is that we're talking about when we talk about something like a synapse. I also want to be very clear about what ontology isn't, or at least shouldn't be. And part of this is a problem of communication when you deal with ontologists. They are a breed apart and they're difficult to deal with. But what it is not is a rigid top-down fixed hierarchy for limiting expression in the neuroscience. That is not what it is. It is rather a way about, not about restricting expression, but how to express meaning clearly and in a machine processable form. It is also not a bottomless resource-eating pit that consumes dollars and returns nothing. It can be that, but it need not be, and I think we've learned some lessons. It is also not a cure-all for all of our problems. It has its place. It doesn't do everything. I don't think it needs to. It is not a completely solved area. There is an active science of ontology, and it is not particularly easy to understand. But what I do want to say is that despite what the ontologists will tell you, where you have ontology and you have things like taxonomies and thesauri and glossaris and controlled vocabulary, and they'll say this is the only thing that matters, the rest of these are unimportant, I don't think so. These are all about expressing meaning in a way that a machine can understand it. And depending on the language that you use and how much time and effort, you can say more or fewer things. That's the point of it. But all of these things have their place. So I take a fairly loose definition of ontology. So why is this important? I wanted to show an example in something that I thought the audience would understand. So this is a Neurolucida file from a tracing program for neurons, and this is representing the dendritic tree of a Purkinje cell. This, however, here, the data, is not a dendrite at all. This is any tree structure. This is a branch structure, and the reason we know that this is not a tree or a road map or a set of blood vessels is right here it says dendrite. Okay? So it's expressing this data type in a universal that we can communicate about. And the interesting thing is that anybody who uses Neurolucida produces something that's under the same level of ontology. Here's an axon, a dendrite, a cell body, and a dendritic spine. So it becomes very easy to combine this data together, and if you want to ask a question, what is the average diameter of a Purkinje neuron, you can ask it, because you can take all that data, add it together, and do the math. Neurolucida doesn't, however, tell you that dendrite belongs to a neuron of a particular type, so it never says that this is a Purkinje cell, or whether this dendrite is neural at all. And if you go and look up dendrite in Wikipedia, you in fact see that there are many different dendrites. It's used in geology, metallurgy, mineralogy. It's a superglue from India and South Asia. You would be surprised anytime you put in a term how many different domains it is used within. When you step outside of your domain and think about that mountain of data, you see again that strings are very ambiguous. So what I like about ontology is ontology really doesn't care about the string itself in all truth. A rose by any other name would smell as sweet. The things about ontology that we care about is that it expresses identity. So entities, a dendrite that belongs to a neuron is different than a dendrite that's a superglue in Asia. They're two different things. They may have the same label, but underneath there is a unique identifier, preferably a meaningless numerical identifier that says, this is the concept. You can call it any number of things you want. It doesn't change the nature of it. The key is the definition. And the definition tends to be of a particular type in different schools, but generally it says there's a genera. This thing is a type of X. And that type of X, again, need not be extremely profound. It's a type of cell. It's a type of anatomical structure. It's a part of a cell. It also may have a set of differentia that distinguish it from something else. So these are a set of Heff's parts or Hasse properties. You can include rules for a definition. You don't need to. And I want to distinguish this from, again, the implementation, whether it's an RDF or OWL or all the different things that you can express it in. Depending on how you express it, you can say more or fewer things, but the basic thing is, what are you talking about? Let us give it a unique name, which is a number, and how do I define it? So why is that very powerful? I'm using myself as an example, right? It's called entity recognition. I want to recognize that thing, that unique thing, and only that thing. So, we heard a little bit about my career, and you can see that here's a set of papers. The Gene Wiki, Ontologies for Neuroscience, three-dimensional electron microscopy, analysis of forgetting pictorial material in amnesic patients, traumatic brain injuries and the goals of care, three-dimensional patterns of enkephalin immune activity in the caudate nucleus. Some are M. Martones or some are M.E. Martones. Can you tell from that which are my papers and which are not my papers? Well, it turns out that even though I spent a lot of time at UCSD, I didn't spend all my time there, and I used to be a neuropsychologist in Boston, and that comes from there. And in fact, I have a cousin, Maryann Martone, who's a medical ethicist who deals with traumatic brain injury, and this M. Martone I have not been able to track down, who also seems to work on wikis and ontologies. But these are the M. Martones that are me. Why is that important? Well, if I identify myself with the TV version of a Social Security number in the US, basically, I'm this number, and I want to use a full URI because this number may be many different things. This URI tells me that this thing actually is a US government label that is myself. And I can have a label, which is Maryann Elizabeth Martone, and I can have all the synonyms I want, and I can have a set of properties. And the main thing is that once I've uniquely identified myself, I can use text mining, I can use 1,000 different algorithms to tell a lot about me. I can do a lot of statistics that say, here's a lot of stuff about me, but I have helped the machine by uniquely identifying myself as myself and nobody else. So we find that ontologies are absolutely essential for the project that we call the Neuroscience Information Framework, because they provide the universals that allow us to query across things, and they provide some way for us to disambiguate, assert identity, and assert things about these things that we're talking about. So NIF has established an ontology, the NIF standard, that essentially has a set of modules. There's organisms, anatomical structures, cells, diseases, subcellular structures. We've actually aggregated these from community ontologies, and I'm going to talk about that in just a moment because I think that's very, very important. And they're very, very simple. They don't say very much. They're just very simple hierarchies. But they serve as very important building blocks for us to say other things about these things. And those things are, as I hope to show you, are going to be very powerful ways of being able to link our data and actually probe our data. So how do we use ontologies in the NIF? Well, first of all, you already saw the query expansion. Here's 3 different databases, one of which is my own, and I want to ask for the gene alpha-synuclein and striatum. I bring back 3 databases with alpha-synuclein, the caudate putamen, the neostriatum, the dorsal striatum of the rodent. Why did I bring those back? Because the NIF ontology says, oh, those are all synonyms of one another. And here's all the synonyms of alpha-synuclein, so if you're going to use, if they use different gene identifiers, you can find them by these names. The problem, of course, is that you would be surprised the number of times, even in neuroscience, that these names aren't unique. So it turns out that PD1, which is a synonym for alpha-synuclein, is also programmed cell death protein and spermatogenesis-associated protein number 2. So even though this can help by pulling data together, there are still times when it doesn't do particularly well. That's when we have to turn to entity mapping. We use entity mapping where we make an explicit declaration that something that's contained in a database is also a member of this particular class, and we use the numeric identifier to say what that class is. So this is the SumsDB that David Van Essen has created and made available through NIF. You'll notice that he's talking about Brodmann.3. When I look at my query statement here, there is no Brodmann.3 in there. It's not a synonym, but there's an explicit mapping between this custom abbreviation and that particular identifier so that we can find it when we're actually looking for it. This also helps us with disambiguating different concepts. So we've also started to knit a lot of knowledge into the neuroscience information framework, because again, if you have this mountain of data, you want to be able to probe it in interesting ways. So we've taken those basic building blocks and we've started to build knowledge and rules into it. So you already saw the GABAergic neuron that we talked about before as being defined by a rule. NIF makes a guess that you as a neuroscientist, when you're looking for GABAergic neuron, is really not looking for the string GABAergic neuron. That's what you get when you go to Google. It's making the assumption that what you really want are members of this class. Because we have that rule stored with that particular class, when you query NIF for GABAergic neuron, it expands out into all the neurons that we know of that use GABA. And we started to expand this beyond simple things like cell classes to sort of fuzzy concepts like adult, increased gene expression. So you might want to ask a question like, what genes are upregulated by drugs of abuse in the adult mouse? Well, there's several concepts in there. There's the adult mouse, there's drugs of abuse, and there's upregulated. NIF says, well, if you're a mouse that's 8 to 12 weeks old, you're an adult. It doesn't matter what you say. Now you might say, well, who are you to say what an adult mouse is? Why do you even want to say what an adult mouse is? You really ought to be looking at these ages. But here again, you have to think about the query interface. If you're going into one database that's about mouse, you might want to give an age range. If you're querying across the internet for adult, how do you know how old an adult ferret is, or an adult chimpanzee, or an adult any other animal? Somebody's got to define that for you. So NIF, being courageous, has said, hey, you know what, adult is really not that useful a concept when it comes to science, but it is a very useful concept for searching for data. So we are just going to tell you, in our view, what are adult animals. We call these things arbitrary and defensible. But again, it shows that there's a definition of this class. In this case, the definition is quantitative instead of qualitative, but it's basically the same thing. So I'm hoping that I showed you that ontologies are fairly essential for making sense out of the mountains of data, and also can form the foundation for really powerful queries where you can actually start mining the data in a language that is familiar to you as a scientist. Now I want to make the case that when you actually build these ontologies, instead of building your own, you really actually want to work with these community ontologies. Now, we heard yesterday that it was a little bit premature for that, but again, I hope I've shown you that a lot of the concepts that are being expressed are fairly universal and generic. To say that we have no commonality and we don't know anything and we have no theory is just as wrong to say that we have it all figured out. Right? The reason why it's so important to use shared building blocks is I've seen it all the time, even in some work that I've done. You have one group that starts from brain and decides to go down to cell. You have another group that starts from cell and goes down to brain. Sometimes they meet in the middle, sometimes they don't. But in fact, there is no explicit relationship between these. You can do automated mapping, you can even do manual mapping, but what often happens is the mapping is incomplete, and sometimes it's not possible because there's all intermediate class. But the main thing is that you don't add to the framework. You've just sort of locked information into your own custom view, and you yourself can't take advantage of all the others' work. Instead, if you take the modular building blocks that have been assembled by NIF, and you say, here's a set of brain regions, here's a set of cell parts, and here's a set of cells, and you start to weave these together, the knowledge base gets enhanced. Also, NIF tries very hard to use community ontologies, so we link ourselves not only to neuroscience, but to the larger life science community where these ontologies are very frequently used. So you therefore make your data much more linkable. You also allow much more powerful things to happen. So for example, you can link to other things. We might talk about the relationship of IP3 to a cell class, but the ontology tells us about its structure and it tells us other properties about it that we can use. We also, when we enrich the knowledge base, allow ourselves to probe it. So here's a page from the Neurolex Wiki, which I'll talk a little bit about in a moment. But essentially, you'll notice it looks like a Wikipedia page, but here are neurons in the dentate gyrus molecular layer, axons in the molecular layer, and dendrites in the molecular layer. These were all computed. They were not asserted. Nobody put those links there. But because there was a rule that was done in a set of consistent ontologies, you could very easily define these and make use of them. Okay? Now you might say, well, everybody has to say the same thing. No, everybody does not have to say the same thing. This is an example that Stephen Larson did for reasoning, but the reasoning example is not that important, but the main thing is that there's a set of statements in here about dopamine. So dopamine is in a ventral tegmental area neuron. Here it says dopamine's a drug. Here it says dopamine's a neurotransmitter. Here it says dopamine's an inhibitory transmitter. Here's dopamine's an excitatory transmitter. We don't have to worry that these are all different dopamines because somebody has told us these are all members of this class. Notice in this case, they've told us two things that are probably consistent with one another. Here they said two things are inconsistent with one another. The way we know they're inconsistent with one another is, again, we have this identity. We don't have to worry about what we're looking at, so we can make evaluations about whether it's correct or not. So these shared ontologies become very powerful building blocks for this framework for communicating in terms of universals. And the thing about it now is that it's a lot easier to get these than it used to be. So the NIF project itself makes available a set of web services that you can go ahead and access our ontologies. You don't need to understand Protégé or other tools. You can just access them directly. The NCBO bioportal is a collection of ontologies, many of which have been mapped to one another that you again can access through services or download them. And as we'll hear a little bit more about tomorrow, the INCF program on ontologies for neural structures, they're also getting into this. So it's much easier to get at them. We've also learned a lot, of course, about, why do we want to use these and why do we want to use them in our own data? Why are these not an afterthought? I thought I would give you a very practical experience that I had with my own database, the Cell Center database. So this has microscopy data, and inside that data we have a lot of segmented objects. We heard yesterday about how hard it is to segment objects and how hard it is to annotate it, and we also heard that we thought it was a little premature to map these to ontologies. Well, here's a typical record of what we get in the Cell Center database. I opened up a file that gave me a list of segmented objects. It was mitochondria 1, Schwann spelled wrong, lower merge, UL cisternae, crop, loop 7, blue, Alex, and lysosome spelled incorrectly. So this was free annotation. Well, it turns out Alex has left. He was a student, so I have really no idea what he was talking about. And the program he used to create the ontology, to create these annotations is obsolete. So I have no way of taking advantage of all the work that he did because I can't look at it. So I maintain that actually using community ontologies for these sort of fairly well-defined concepts is very powerful, and it's not that difficult. So for example, we've started to add into a lot of our segmentation tools these ontology services so that when you annotate, you're actually creating instances of the ontology. And you're using a coherent model in which to express relationships. That also helps us because you saw the size of those data sets yesterday. You saw how big those EM data sets are. By saying that this is an instance of endoplasmic reticulum and this is an instance of dendritic spine, I can use that knowledge to actually hone in on areas that satisfy a condition. So here the query is, where in that 2 terabytes of data are there instances of spines, concept 1, with membrane-bound organelles? I can go and find that information very easily because I can take advantage of the knowledge built into the ontology. We've also made it fairly easy for people to look at it. We think one of the mistakes of the ontology community was that they made it a little bit too difficult and they started at too high a level. We actually believe that it should be easy for neuroscientists to say what concepts they need to describe their data, provide some standard way of describing those concepts, get an identity from it, get a URI, and then leave ontologists and other engineers to build in that complexity later. So we started the NeuroLex Wiki. This is a project of Stephen Larson, and there's a demo outside to make it very easy for people to be able to add things so that if there isn't a concept that you need, you can go ahead and add it. So I don't want to leave the impression that we know everything about making that linked data cloud work, that we know everything about how to make our data available. We had the printing press in the 1300s. We've had the web of data for a few years. We haven't had it very long. We've never exposed our data this way. We don't know all of the things. And I would say right now that effective data sharing is still an act of will. But just because we don't know everything, I don't want to leave you the impression that we know nothing. That through our experiences in NIF, we have found that there are several things that you can do as a resource provider and as a researcher to start to adapt your practices to recognize that your database is not a silo, that it is not self-contained, that it should be exposing its content to the world for it to use the way that they want to use it. Of course, the most important thing is that you should register your resource with NIF. And we have a little registry button. I'll be happy to go through and show you. And I want to thank our colleagues in Japan because they have this proudly displayed on their website. But there's a few other things. And right now, I sort of link these together under this concept of mindfulness. We may not know everything about it, but we do know that if you remember that your resource is contributing to a global federation, and that you link to shared ontology identifiers where it's possible, that you remember that stable and unique identifiers for data are critical. I can't tell you the number of times in NIF that we have linked to a database, they take it down, they reuse the identifiers, but now those identifiers all point to different things. Microarray chip manufacturers do the same thing. A new version of the chip, the probe IDs now point to different things. That's the kiss of death on the web of data. This thing should be resolvable to something, and it should stay, and it should be persistent. These explicit semantics, and these are different types of resources. They're models, they're XML files and things. They need to go in there. For researchers, you need to be mindful that when you are publishing data, no matter where it is, in a paper or otherwise, that it is to be consumed by machines and not just your colleagues. There's a couple of very simple things to do, to put accession numbers for genes and not strings, because strings can be ambiguous. Actually tell us what species it is, and preferably use an NCBI taxonomy ID. For reagents, start putting in catalog numbers. I didn't talk about supplemental data, but I could give a whole talk on that. So, there are some hopeful signs that, first of all, the means for sharing data on the web is becoming more routine. It's not difficult. NIF has a lot of tools for exposing the content of your relational data, XML data, RDF data, whatever it is, through an interface. But we also, I think, have a recognition by funding agencies and international agencies that this is necessary. There's growing recognition for the role of standards and curation, and we have organizations that can help. I've talked about NIF. We have the NITRIC activity, which is a software tools. We've heard about the software tool repository for INCF. We have the INCF itself, which I think can start to educate neuroscientists on what's necessary. It's established a very effective model for bringing together stakeholders to define what's necessary for interoperation, and it's implementing structures and procedures for developing neuroscience resources within a framework. So one thing I want to emphasize is that a lot of times when people hear standards and they hear about the things they're doing, they think there's only one, that we're sort of clamping down expression and there's only one way to do things. And this is the Highlander, in the end there can be only one, and fight to the death. But we know that there are a lot of standards, and we know that because of the nature of the web and everything else, that multiple efforts are always going to be underway simultaneously. Sometimes they're launched without knowledge of others, but other times you got funds by denouncing the other's efforts, because you can always do things better the second time. So I think a model of competition, cooperation, and coordination really needs to be established in order to make this move forward faster. We always need to consider when we're making a resource, do we need to build a better mousetrap? Or will the current one do, and can we just make that better? Sometimes the answer is yes, sometimes it's no. But I do know that if we don't cooperate and coordinate, we'll keep covering the same territory over and over again. We will keep having the same 300 neurons represented in the database and never be able to branch out. So When INCF is developing standards, and ontologies are one of those components, they are ways of helping us exchange. It's about expressing data in a way so that it can be exchanged with something else, so that you don't have to do this exhausting mapping. If you're building a new resource and you can use these standards, great. There's very little work to do. If you already have an existing resource, you can use the tools established by INCF to transform into other things. And you saw that out there, those of you who have seen the Voxhome space that has been done. It's a way of translating amongst coordinate systems. It doesn't tell you that you can use one or another. So standards make it easier to build interoperable and integratable data sources. So in conclusion, again, I want to emphasize, neural space is vast, and nobody can cover it all. Okay, it is going to require global cooperation and mindfulness, again, that when you are producing your data, you should be producing it in a way that helps us fill in the space. We need to learn new ways of communicating, recognizing the globalization of data. It's no longer publishing it for you and your colleagues. Decisions can be made at the outset that will make it easier or harder. And I want to leave the fact that we get very angry at commercial providers who don't make their products interoperable. We don't like it that we can't use charger A in cell phone B. We don't like it that we can't use microscope lens in microscope B. Yet when it comes time to make our data interoperable, we say, I'm sorry, we don't think that that's our job, and we don't think it's important. And again, just because we don't know everything doesn't mean we know nothing. I also want people to remember that what we're talking about are not false dichotomies. If I hear that it's all going to be automated, it's all going to be human, it's all going to be top-down versus bottom-up, OWL versus RDF, researchers versus curators, it's everybody and everything. Neural space is vast. Thank you. My colleagues.
Thanks, Maryann, for a wonderful presentation, and right on time. We have time for discussion.
Yes.
That was very nice. I guess one of the problems is always convincing researchers to do this.
Yes.
So could you comment on perhaps what sort of carrots, as it were, researchers could be offered on the one hand, and perhaps what sort of sticks might be offered on the other, perhaps from the journal editors and things like that. Why are journals so reluctant to enforce this on people? Is it just that they're scared that they will lose people submitting their lovely experiments and things like that?
I think that there's a part of that. I think that at some level, my more cynical friends say that journals don't want to make this easier because then you don't have to buy their journal, right? So, you want to be able to use their search strategies. I think the issue of carrots and sticks, it has been discussed. Right now, we don't have funding agencies that enforce this. We don't have journals. One of the reasons that NIF is really trying to emphasize extremely simple things that you can do is that at least some of the reluctance I've heard is NIH doesn't like to pay. I'm using just the US because that's what I know. They don't like to pay for databases anyway. They certainly don't want to pay for transforming databases and retrofitting them and so on. So if you tell them that you're going to have to do all of these things, they'll say forget it. If you tell them, however, that really, listen, you're only talking about 4 or 5 things here. I have heard it considered at NIH that they are willing to beef up their data sharing policy that says if you're going to create a resource, this is what you need to do. There are program officers and others who are willing to put those in if we can specify it exactly. And whereas before you might have somebody say, oh, well, you've got to do it RDF, or you've got to do it this way, you've got to do it that way, I think that's at the wrong level. I think it's at the level of saying, listen, don't retire your identifiers and make them point to other things. I mean, again, if there's a shared ontology, you should use it. If there's not a shared ontology, don't. There are simple things that you can do to be mindful of the fact that your data is going to be produced. For journal editors, it's the same way. We've been trying to convince them for a long time. There's a little bit more momentum now because I think researchers are finally starting to realize how much time they waste searching for things and looking for things that they should be able to find, but that they can't. And we've had it, when I've started to present this to researchers, they said, listen, if you tell us what to do, we're happy to do it. There is going to be a certain set that just aren't interested and they don't care. And I've tended to say, well, you know what, there's not a whole lot I can do about them right now until somebody's willing to enforce it. But for these others, I want to give them a very nice little set of rules that they can say, I can say, here's things that we found to be very effective. And they're not horrible. They're not a horrible thing to do. Reference your reagent inside of your paper. I had this wonderful experience this summer with a high school student who I had working on curation of data, and his mother was a researcher. And after he finished curating data from a paper, he went to his mother and he said, when you publish your data, you make sure you put all your reagents in that paper. I don't want to have to go looking in 5, 6 papers to find it, you know. And so I think that, you know, I've always been wanting to go after the people who are willing, but to be able to formulate things in a way that they actually can implement. So.
One more question, please.
Just a short question. Last year there was in Pilsen there was a guy from Google. I can't remember the name actually, and he was explaining how to get to the deep web. So I was wondering how much could is it possible to involve companies like Google in a project like this? Because after all, they want to make all the knowledge of the world accessible.
Well, we actually had a good conversation with Google right afterwards because Jeff Greta had presented NIF, and he basically said, you know, bless you, we're happy you're working on it. Google's not interested because it's not $1 billion market. We're not selling automobiles. But we do think that they have a very important role to play, and we're hoping with some of these new, you know, search engines and things that are out there that do rely a little bit more on the structured data, that NIF or other places can make it available so that you can use it. You know, we don't feel that you have to use one search engine or another. It's whatever's effective. We've also had a lot of worry, for example, you know, from program officers. I have a particularly worrisome hand-wringing program officer, and she's like, well, what if Google, you know, beats us on this? And I'm like, well, you know, then I say, happy for us, we'll go back and do other things. But I think the lasting legacy, again, of things like NIF is neuroscientists, we know what's needed to use data. The search engine companies, Microsoft, they don't know what to do with data. They don't really understand data beyond the level of sequence. They don't really even understand sequence data. We have to be a little bit better with our data, and then it can be used by many different places. But I think the more we can involve companies, the more we can get their really fancy visualization tools, their fancy algorithms, the more we make it easy for them to use our data, the better off we will be.
Okay, thanks again for a terrific presentation.